Our detectives are uncovering the 3 ways the complement system is activated: classical, lectin, and alternative. Each pathway starts the investigation differently, setting the stage for what happens next.
Each case has its different clues. Similarly, our immune system has 3 different pathways that can activate the complement cascade. Our first clue today is DNA evidence that'll lead us to the culprit, hair. The culprit-specific DNA is just like the pathogen's specific identity in the activation of the classical cascade. For this to occur, antibodies must recognize specific antigens on the surface of the pathogen, making it identifiable to the immune system, just like how our forensic scientists will use the DNA in this hair to identify the culprit. Our next clue is fingerprints. The specific pattern in fingerprints are just like the specific pattern recognition that occur in the lectin pathway. In this, mannose binding lectin binds to specific carbohydrate patterns on the surface of a pathogen. The MBL protein acts kind of like detectives that are searching for a specific pattern that will match and trigger the complement cascade, just like how fingerprints will match a suspect. Our final clue is tire marks. We can't conclude that these tire marks are specific to our crime, just like the alternative pathway, which isn't activated by anything specific. It spontaneously activates in the presence of a pathogen. While these tire marks won't lead us straight to our culprit, they'll add to our case and allow us to continue our search, just like how the alternative pathway will add to our immune response.
During activation of the complement system, C3 convertase cleaves C3 into 2 separate parts, C3A and C3B. C3A is responsible for promoting inflammation, while C3B marks pathogens for destruction. Just like finding a critical piece of evidence, the cleavage of C3 into C3A and C3B helps us understand the immune system.
Pathogen X, but it won't be around for long. Once the immune system kicks in, let's see how the 3 consequences of complement action take down this invader. First is inflammation, like a crime scene response unit. Inflammation seals off the area, calling in backup. The body's immune system is now on high alert, ready to respond. Next, we have opsonization, the body's tagging system. Just like how forensic experts tag evidence, opsonins mark pathogen X for destruction, making sure immune cells know exactly where to strike. Pathogen's defense, creating a breach in the membrane. With inflammation marking the site, opsonization attacking the invader, and MAC formation delivering the final blow, the case is closed and pathogen X is no more. The immune system prevails again.
Here's how the 3-3-3 rule played out in our investigation. There are 3 ways to cleave: classical, lectin, and alternative pathways activate the complement system. The cleavage of C3, splitting C3 into C3A and C3B, provides crucial evidence. 3 consequences: inflammation, opsonization, and the membrane attack complex formation, resolves the case. And that's how our CSI team cracks a case of the complement system, ensuring your immune system stays protected from invaders. Case closed.